Welcome to the Deep Dive. Today we are looking at a stack of incredibly dense bioengineering research, specifically a massive July 2026 breakthrough published in Cell Reports Medicine.
Yeah, it's a fascinating paper.
It really is. And our mission today, for you listening, is to uncover how scientists are fundamentally changing the way we fight cancer. I mean, think about it like this. If you were about to fly across the ocean in a highly experimental aircraft, you really hope the pilot didn't just, skim an instruction manual.
Right, absolutely.
You'd hope they spent hundreds of hours in a state-of-the-art flight simulator.
You would demand a simulator that perfectly mimics reality.
Yeah.
I mean, one that throws every conceivable crosswind, sudden pressure drop, catastrophic system failure at the pilot, all while everyone is safely on the ground.
Exactly.
You need to know how the plane reacts to chaos before you are 30,000 feet in the air.
Because you absolutely do not want to test how the plane handles a failure when lives are actively on the line. But if we take a step back and look at standard oncology, for decades, that is unfortunately how we have had to approach treating colorectal cancer.
Yeah, it is a sobering comparison, but the mechanism of treatment has largely relied on that kind of high-stakes trial and error.
It's brutal.
It is. We give a patient a heavy, highly toxic chemotherapy regimen, and then we simply have to wait months to see if that specific chemical cocktail actually works for their unique biology. We're essentially testing the experimental plane while it is already in flight.
Which brings us to the core mission of today's deep dive. We're unpacking an invention that feels ripped out of a science fiction novel, but it is actively saving lives right now. It's a bioengineering marvel called the vascularized colorectal tumoroid-on-a-chip.
It's quite a mouthful, but the technology is incredible.
Totally. So in plain terms, scientists have figured out how to build a tiny, living, pumping model of a patient-specific colon cancer, completely outside their body. And they're using this tiny piece of plastic as a perfect miniature flight simulator to crash different highly toxic drugs, predicting exactly which one will actually save that patient's life.
The operative concept there is getting as close to perfect reality as physically possible. To understand why this specific microchip is such a monumental leap forward, we really have to talk about what previous laboratory models completely missed.
Right.
We have to look at the tumor's infrastructure. Basically, we have to talk about the plumbing.
Okay, let's lay the groundwork here for everyone. In previous deep dives, we've talked about organoids. These are those fascinating 3D clusters of cells grown in a petri dish that mimic human organs, or, in this case, tumors.
Right, the 3D models.
Yeah. So if we already had these 3D tumor models sitting in labs, why couldn't scientists just drop the cancer drugs onto them and see if the cells die?
Well, a tumor, especially an aggressive colorectal cancer tumor, is not just a static lump of rogue cells sitting in a void. It's a highly complex, dynamic, and aggressively expanding ecosystem.
It's alive.
Exactly. And to expand, it is highly vascularized, meaning it relies heavily on a dense, intricate network of blood vessels to deliver massive amounts of oxygen and nutrients and, to carry away the cellular waste. It essentially hijacks the body to build its own private supply chain.
Oh, wow. So to test a drug that is delivered intravenously, you just can't ignore the highway system it takes to get to the destination.
You really can't.
It's like testing a drug by dropping it directly onto a naked tumor in a dish is like testing how waterproof a pair of hiking boots are by just dropping water directly onto the inner fleece lining.
That's a great way to put it.
It completely ignores the protective outer shell and the actual journey the water takes.
That analogy hits the nail on the head. Standard 3D tumor models entirely lack this vascular network. They have zero blood vessels. So researchers logically thought, well, they could solve this by simply growing normal, healthy human blood vessel cells right alongside the tumor in the lab.
Which seems like a logical next step, right?
It does. They usually use standard ones called HUVECs, which stands for human umbilical vein endothelial cells. But that attempt introduced a massive biological roadblock known as the shunt problem.
The shunt problem. Okay, break down the mechanics of that for us. Why do healthy blood vessels fail to simulate the real thing?
Because the plumbing of a real aggressive tumor operates under completely different laws of physics compared to the plumbing in the rest of your body. Normal blood vessels are organized, orderly, and relatively watertight. They're designed to maintain steady blood pressure.
Right, like normal pipes.
Exactly. Tumor blood vessels, on the other hand, are uniquely chaotic. They are structurally deformed, they branch irregularly, they twist violently, and critically, they are incredibly permeable. I mean, they are riddled with microscopic holes.
So instead of a functional highway system, the tumor's infrastructure is more like a massive swamp filled with microscopic sinkholes.
A swamp is the perfect way to visualize it. And that gritty, messy reality completely changes the internal fluid dynamics and the hydrostatic pressure of the tumor's microenvironment.
How so?
Well, think of a normal blood vessel like a water slide. The fluid rushes straight through, maintaining pressure, delivering the drug efficiently to the target. But in the swamp of a tumor, as the drug travels through, the pressure drops instantly.
Because it's all leaking out.
Exactly. The fluid is just leaking out through those sinkholes into the surrounding tissue.
It never builds up the momentum or the concentration needed to actually penetrate the hard, dense outer fortress of the tumor itself.
Which means if you just put normal, healthy, watertight blood vessels next to a tumor in a lab, your drug predictions are going to be completely inaccurate. You've built a flight simulator, but you program the weather to be perfectly clear and sunny when the reality inside the patient's body is a Category 5 hurricane.
But this raises a seemingly impossible engineering challenge. If tumor blood vessels are essentially these rogue, chaotic, leaking versions of normal plumbing, how do you recreate that exact, specific mess in a sterile, highly controlled laboratory?
It's incredibly difficult.
You can't just politely ask normal, healthy cells to malfunction and build a swamp, right?
No, you can't. And that specific hurdle is where the researchers achieved their biggest biological breakthrough. They realized they couldn't just use normal cells, but they also couldn't reliably extract the incredibly fragile tumor blood vessels directly from patients.
So what did they do?
They had to take healthy, functioning blood vessel cells and actively educate them to act like cancer cells. They literally had to teach them to go rogue.
Educate them. Okay, I love this terminology. How exactly do you brainwash a healthy biological cell into building a chaotic swamp?
It started with a massive computational beta hunt. The researchers dove into single-cell sequencing databases, specifically two massive datasets known as GSE 144735 and GSE 132465.
Okay, that's a lot of letters and numbers. What are those?
Essentially, these databases contain the microscopic genetic blueprints of thousands upon thousands of individual cells. They ran a deep comparative analysis between normal vascular endothelial cells and colorectal cancer vascular endothelial cells.
They were basically looking for the molecular needle in the haystack, the specific instruction that tells the tumor cells to build leaky pipes.
Yes. And when they intersected the data from both of those massive databases, they found one specific protein that was massively, disproportionately overexpressed in the tumor vessels compared to the normal ones. It is a protein called IGF-BP5.
IGF-BP5. So this single protein is the bad influence. It's the corrupting syllabus that the cancer uses to brainwash healthy infrastructure.
Precisely.
But how does one protein actually change the physical architecture of a blood vessel?
Well, proteins act like instruction manuals for the internal machinery of a cell. To test this theory, the scientists took those normal, healthy HUVECs, the standard watertight blood vessel cells, and bathed them in a heavy concentration of this IGF-BP5 protein.
How heavy are we talking?
Specifically, 50 nanograms per milliliter. When these healthy cells are exposed to this massive dose, the protein binds to their outer receptors and completely overrides their normal biological blueprint.
So instead of receiving the standard signal to, build a smooth watertight tube, their internal communication network just starts misfiring.
Their internal growth signals become incredibly erratic, and the results were visually astonishing. Within just 4 to 24 hours of exposure, these educated cells completely changed how they physically grew.
What did it look like?
Under the microscope, the researchers saw them forming highly chaotic networks. They had vastly more junctions, extremely short and stunted vessel lengths, highly irregular diameters.
Bulging in weird places?
Yeah, some thick and swollen, some incredibly thin, and a huge number of discontinuous dead-end branches.
They successfully grew the swamp. But seeing a tangled mess under a microscope is one thing. How did they actually prove that these new educated vessels were leaking pressure the way a real tumor does? Because morphology, how something looks, doesn't always guarantee function.
You're completely right. To prove the physics were correct, they ran a brilliant diagnostic test. They pumped a special green fluorescent fluid, a polymer called dextrin, through these newly educated vessels.
Okay, glowing green fluid.
Yeah. Under fluorescent microscopy, they could actively watch the bright green fluid rushing through the tubes and then rapidly leaking out of the vessel walls and bleeding into the surrounding dark space.
Oh, wow. A visible, undeniable proof of permeability.
And they took it even further. They ran tiny blue fluorescent microbeads through the network. In a healthy, normal vessel, those blue beads would just shoot straight through like cars on a highway.
But in the swamp.
In these educated IGF-BP5-soaked vessels, the flow rate dropped off a cliff because of the irregular, warped pipe sizes. After 2 hours, they found massive amounts of these solid blue beads had just drifted entirely outside of the vascular network.
Just trapped in the surrounding tissue.
Exactly. They had perfectly mimicked the extreme permeability and the chaotic, low-pressure fluid dynamics of real tumor vasculature.
Okay, let's do a quick recap to anchor where we are for everyone listening, because the density of this science is incredible. So researchers realized they needed the messy, leaky plumbing of a real tumor to properly test cancer drugs.
Right.
By analyzing massive genetic databases, they found a single protein, IGF-BP5, that acts as a corrupting blueprint. By marinating healthy blood vessels in this protein, they forced them to misfire and physically build the exact kind of leaky, chaotic swamp that protects a real tumor.
That is the perfect summary of the biological foundation. But having a puddle of rogue cells swimming in a petri dish isn't a simulator.
Fair point.
To actually test a systemic drug, we need to force fluid through those vessels and route them directly into a living, beating piece of a patient's actual tumor. And that is where the biology experiment transitions into a hardcore physical engineering problem.
We move to the realm of microfluidics.
Yeah.
How do you physically combine this microscopic swamp with a piece of human cancer in a way that you can actually control and measure?
So they engineered a highly specialized microfluidic chip out of a material called PDMS. PDMS is a flexible, highly transparent, and perfectly biocompatible polymer.
Basically a clear, rubbery, high-tech plastic block.
Exactly, and it's small enough to easily fit in the palm of your hand.
So you have this clear plastic block. What does the internal architecture actually look like?
Well, in the very center of this chip, there is a tiny hexagonal central chamber. That is the throne where the patient's specific colorectal tumoroid is placed.
Okay, the VIP section for the tumor.
Yeah. And surrounding that central chamber are channels where the educated blood vessels are going to grow. But here's the tricky part. If you just inject a mixture of cells and biological gel into those channels, you just end up with a solid, useless block of jelly.
Right, because you need hollow tubes running through the center of that gel so the artificial blood can actually flow.
Exactly. You need to lay the actual piping before the concrete sets, so to speak.
How on earth do you carve a hollow microscopic tunnel through liquid gel?
They used 250 micrometer acupuncture needles.
Wait, 250 micrometers? That is roughly the thickness of 2 or 3 human hairs. You're telling me they are threading literal, physical acupuncture needles through this tiny plastic chip?
It requires immense precision. They slide these microscopic metal needles horizontally through the channels of the chip. Once the needles are in place acting as placeholders, they inject a mixture of the educated blood vessel cells and a fibrin gel around the needles.
Okay, and then?
They let the entire chip sit for 30 minutes at exactly 37 degrees Celsius, which is normal human body temperature. Once the gel firmly solidifies, they incredibly carefully pull the metal acupuncture needles out.
That is just brilliant. So the needles slide out and leave behind perfect microscopic hollow tunnels running right through the middle of the solid gel.
And because biology is amazing, the educated blood vessel cells naturally migrate to line the walls of those hollow tunnels. From there, they hook the edges of the tunnels up to a precision syringe pump.
To simulate a heartbeat.
Sort of. This pump constantly, gently pushes liquid culture medium, which acts as the artificial blood, through the chip at a highly controlled rate of 10 microliters per hour.
10 microliters an hour. That's barely even a trickle. But I imagine it perfectly mimics the slow, constant, creeping flow of capillaries deep inside the human body.
It really does.
But wait, I have to push back on a major biological variable here.
Yeah.
If you take an aggressive piece of a patient's colon cancer, and you take these engineered blood vessel cells and you jam them together into this tiny shared plastic apartment, don't they require totally different environments to survive?
You would think so, yes.
Doesn't the chemical food that keeps a tumor thriving absolutely poison a blood vessel?
That exact compatibility issue is a massive hurdle in co-culturing any two distinct types of cells. The researchers spent a significant amount of time meticulously testing varying ratios of tumor culture medium versus endothelial culture medium.
Trying to find a compromise.
Right, and what they discovered was that a perfectly balanced 1.1 mix was the absolute sweet spot for survival.
A 50-50 split of the nutrients. But did sharing the food supply accidentally change the cells? Like, did the tumor start acting less like a tumor?
They had to verify that, of course. They checked the specific biological markers of the cells after they were mixed. They looked for CD31, which is essentially a molecular name tag that proves a cell is part of a blood vessel.
Got it.
And they looked at markers like Ki67, which lights up when a cancer cell is actively dividing and multiplying. The cells perfectly maintained their true identities in this mixed environment.
So they successfully coexisted.
Actually, it goes way beyond coexisting. Here is where we see the sheer power of this model. The tumors didn't just survive in this chip. When connected to the educated blood vessels, the tumors actually grew significantly larger and more aggressively than when they were grown all by themselves in a traditional vessel-free dish.
Oh, wow, because they finally had a working supply chain. The plumbing was actively feeding them.
Exactly. The educated blood vessels actively transported the nutrients right to the tumor's doorstep, sometimes even physically invading the hexagonal tumor chamber to feed it directly.
Wait, invading it?
Yeah, it mimics a process called angiogenesis that happens in the human body, where tumors force the creation of new blood vessels. Remember those blue fluorescent beads we talked about earlier?
Like one's getting stuck in the swamp.
In the live chip, they traced those solid beads flowing out of the leaky vessels and washing right up against the edge of the living tumor cells.
Okay, so they have successfully engineered this incredible, biologically accurate pumping micromodel of a swamp-like tumor environment. But let's elevate this from the laboratory to the real world. For a listener at home, does this tiny piece of plastic actually have the power to save human lives?
That's the real question.
Can it really predict if a massive dose of chemotherapy is going to work on a patient?
This brings us to the ultimate stakes of the paper, the clinical validation phase. The researchers took living tumor samples from 22 real human patients who were actively undergoing a specific heavy-duty treatment regimen for colorectal cancer.
What was the treatment?
The standard treatment was a chemotherapy cocktail called FOLFOX, combined with a targeted drug called bevacizumab.
Okay, so the doctors are treating the actual patients in the hospital, and simultaneously, the researchers are treating their biological twins, the tiny living chips, in the laboratory simulator.
To see if the simulator can predict the future. And the data from one specific individual listed as patient P11 perfectly illustrates why this complex biological engineering is so utterly vital.
What happened with P11?
In the actual hospital, patient P11 was suffering terribly. Their liver metastases were actively growing larger despite the heavy chemo, and their blood tests showed alarming dramatic spikes in a marker called CEA.
Let's define CEA for a moment so we grasp the severity here.
CEA stands for carcinoembryonic antigen. Think of it as chemical exhaust. When colon cancer cells are aggressively multiplying and thriving, they shed this specific protein directly into the bloodstream.
So a spiking CEA level means the cancer is winning.
Yes. So for patient P11, the FOLFOX and bevacizumab combination was completely failing. The tumor was highly resistant. The chemotherapy was essentially doing nothing but causing the patient immense physical suffering.
Absolute clinical resistance. Now let's look at what happened in the laboratory simulator. What did the chip show?
The researchers put patient P11 cells into two different types of chips. One was the old school model, populated with normal, healthy, uneducated blood vessels. The other was the new optimized chip, featuring the leaky, chaotic, IGFBP5 educated swamp vessels.
Let me guess. The old chip completely failed to predict the resistance.
The old chip got it disastrously wrong. In the old chip, the drugs utilized those nice, pristine, watertight blood vessels like a high-speed water slide. The chemotherapy blasted straight through the pipes, maintained perfect pressure, penetrated the tumor, and killed it. Yeah, the old chip showed that the drugs were working beautifully. It was a massive false positive for the drug's efficacy.
Wow. If an oncologist had relied on that old, clean chip, they would have been falsely reassured. I mean, they would have kept giving patient P 11 months of toxic, debilitating chemo that was never, ever going to work in reality.
But in the newly optimized chip, the one with the messy, realistic, educated plumbing, The results were entirely different. As the drug pumped through, it hit the sinkholes. It leaked out into the surrounding gel, losing all of its pressure and concentration.
And the tumor.
The tumoroid inside the hexagonal chamber kept right on growing, completely brushing off the diluted drugs. The optimized chip perfectly, flawlessly predicted the grim reality that patient P11 was experiencing in the clinic.
That is the ultimate aha moment right there. The false positive in the old chip was basically an illusion caused by perfect plumbing. By recreating the chaotic biological swamp, they finally built a simulator that accounts for how hard it really is for a drug to physically penetrate the tumor's fortress.
Exactly. And when they expanded this test to all 22 patients, the optimized educated chip had an astonishing 86.36% accuracy rate in perfectly matching the actual clinical outcomes of those human beings.
86%.
And its specificity, meaning its ability to correctly identify when a drug would actively work without failing, was over 90%. By engineering the biological chaos, they drastically reduced the false negative problem that has plagued customized cancer research for years.
So if we connect this to the bigger picture for you listening, this chip could literally prevent us from giving a patient months of nauseating, immune-destroying chemotherapy that isn't even going to touch their cancer. I mean, it cuts out the horrific trial and error phase.
It fundamentally spares the patient immense physical suffering and prevents the catastrophic loss of precious time. But the implications go even further.
How so?
Because once you use the simulator to quickly prove a standard treatment isn't going to work, you can use that same chip proactively, aggressively to find a completely different treatment that will.
Expanding the arsenal. Okay, so how do they use the chip to go on the offensive when standard chemo fails?
We can look at another compelling case study from the paper, patient P15. This patient was in an incredibly tough spot. They had already endured multiple rounds of different aggressive, broad-spectrum chemotherapies, and yet their lung metastases just kept rapidly growing.
So they were effectively running out of standard first-line options.
Yeah, the terrifying back-against-the-wall scenario that every single patient and family dreads.
Right.
In those scenarios, oncologists often look to third-line treatments. Traditional chemo. These are highly specific drugs designed to attack the blood vessels, basically choking off the tumor's blood supply.
Oh, starving the swamp.
Exactly. The problem is there is a large menu of these targeted drugs available, and currently there are 0 reliable biological markers to tell a doctor which specific drug will work for which specific patient. It's normally just an educated guessing game.
But with the microfluidic chip, it doesn't have to be a guess. You can just run the simulation.
Right. They took patient P15's tumoroid and hooked it up to the optimized educated plumbing inside the chip. Then they systematically tested a whole menu of different small molecule targeted drugs.
Which ones did they try?
They ran the simulator with apatinib, then sunitinib, fruquintinib, sorafenib, and regorafenib. They ran 5 completely different flight simulations.
And what did the simulator reveal?
The chip clearly and undeniably identified that patient P15's specific tumor and vascular network were highly sensitive to one single drug out of the lineup, regorafenib. In that specific simulation, the blood vessels violently withered, the supply chain collapsed, and the tumor shrank.
And the ultimate question, did they give regorafenib to the actual human patient?
They did. Based on the data, patient P15 was given regorafenib in the clinic, and shortly after, their clinical imaging showed a verified partial response. Their lung metastases significantly shrank. The chip acted as a personalized, perfectly accurate crystal ball.
That is simply incredible. We are talking about shifting the entire field of oncology from a brutal game of broad statistics to a game of absolute personalized precision. Let's take a final step back and summarize this incredible journey we've just been on so it really sticks with you.
The paradigm shift here really cannot be overstated.
The scientists finally realized that to test cancer drugs accurately outside the body, you can't just look at the tumor. You desperately need the plumbing. But you need the chaotic, leaky, twisted plumbing of a real cancer environment.
The swamp.
Exactly, the swamp. By digging through massive genetic databases, they identified a single corrupting protein, IGFBP5. By drowning healthy blood vessels in this protein, they essentially overrode their instruction manuals, forcing them to physically build the exact leaky environment that protects real tumors.
And by merging biology with microfluidic engineering, literally using microscopic acupuncture needles to carve hollow tunnels and clear plastic chips, they built a tiny, pumping, living ecosystem that houses both this rogue plumbing and a piece of the patient's actual colon cancer.
And when put to the ultimate test against real human patients, this beautifully chaotic chip achieved over 86% accuracy in predicting exactly which toxic drugs would fail and which targeted drugs would actually shrink the tumors. It completely solved the dangerous false positive problem of older, perfect models.
It is a profound masterclass in using cutting-edge bioengineering to perfectly mimic the grim, messy reality of biology, all in the name of saving lives.
So for you listening, if you or someone you love ever faces a diagnosis like this in the future, this deep dive represents a reality that is vastly less terrifying. It represents a near future where the brutal, painful trial and error phase of oncology is entirely eliminated.
Which is life-changing.
It really is. Your specific cancer is tested, fought, and defeated on a tiny plastic chip, safely on a laboratory bench, before a single drop of medicine ever enters your veins.
It is the absolute pinnacle of personalized medicine. We are finally truly testing the airplane in the simulator, completely avoiding the risks of testing it in the sky.
And as we wrap up, I want to leave you with one final mind-bending question to mull over. We just spent this entire deep dive exploring how scientists discovered they could educate normal, healthy cells to become chaotic and tumor-like just by exposing them to one specific protein. But what if we could do the exact reverse?
Oh, that's an interesting thought.
Right. Could we eventually discover a different protein or engineer a drug that re-educates the tumor's rogue blood vessels inside the patient's body? What if we could force the swamp to behave normally again, completely cutting off the cancer's life support from the inside out?
Using the tumor's own infrastructure as a weapon against it. That is a fascinating, highly plausible question for the very next frontier of cancer research.
It really is. Thank you so much for joining us on this deep dive. Stay curious, keep questioning the mechanics of the world around you, and we will see you next time.